Imagine for a second, you're sitting in this sleek, brightly lit dermatologist's chair. You know the kind.
Oh, yeah, the really intimidating one.
Exactly. And you're about to have a highly purified liquid, which is actually extracted directly from salmon sperm, injected right into your face.
Which sounds completely wild when you just say it out loud like that.
It really does. But I mean, you're probably paying anywhere from, what, $500 to $2,000 for this single session. And you're doing it because this treatment injectable polynucleotides, or PDRN as we'll call it today, is widely considered an absolute miracle for skin rejuvenation.
Yeah, it's pretty much the gold standard right now.
Right. But here is where the story takes a massive turn. The secret to why this $1,000 treatment actually works, like why it does anything at all, isn't just about the DNA itself.
Not at all.
No, it's entirely dependent on this invisible biological tug-of-war happening right under your skin.
Right, I mean, the real mechanism relies on a completely natural shredding enzyme in your body that, well, it actually views this incredibly expensive treatment as a threat, and it is constantly, aggressively trying to destroy it.
And that conflict is exactly what we are diving into today. We've got a fascinating stack of sources for you for this deep dive. We're pulling from cutting-edge biotech patents, clinical cosmetic guidelines, and some incredibly dense molecular biology research papers.
Very dense, lots of charts.
So many charts. But our mission today is to unpack how your body's native tissue deoxyribonuclease enzymes, which thankfully we're just going to call DNase, how they completely control the gradual breakdown and the absorption of these injected polynucleotides.
It's honestly a brilliant story. It's about how our body's ancient defense mechanisms are interacting with, very modern cosmetic science. But to really grasp what happens when that needle goes in, we have to start by understanding the native environment of the skin itself.
Right, like what's actually going on down there?
Exactly. We have to look at what these shredding enzymes, these DNase, are actually doing on just a random Tuesday.
Well, let's start right there with the baseline, because before someone intentionally injects salmon DNA into their cheeks, what is DNase doing in our bodies naturally?
So at a molecular level, you can kind of think of DNase enzymes as biological scissors. In textbook terms, the sources describe them as glycoprotein endonucleases.
Okay, which means what exactly?
Right. Functionally, that just means their sole purpose in life is to hunt down the chemical bonds that hold DNA together and just snip them apart. They are essentially your body's microscopic cleanup crew.
Because our cells are dying all the time, right? Just from normal, everyday wear and tear.
Exactly. Every single minute of the day, your cells are dying off. Sometimes it's a very neat, biologically programmed process, which is called apoptosis.
The clean version.
Right, the clean version. But other times it's messy, unprogrammed damage from the environment, UV rays, pollution, and we call that necrosis. Either way, when a cell bursts, it spills all of its internal DNA out into the surrounding tissue and into the bloodstream.
Gross.
Yeah, it's cellular. In the medical literature, they call this extracellular DNA, or eDNA.
And I'm guessing stray DNA just floating around in the blood is a bad thing.
Oh, it's a massive red flag. If that eDNA isn't cleared out rapidly, your immune system spots it and assumes you are under an active viral or bacterial attack.
Oh, wow. So the immune system doesn't even recognize it as your own DNA.
Nope. It just sees raw genetic material where it absolutely shouldn't be. And so it triggers this severe systemic inflammatory response.
So these DNase enzymes are essentially they're like biological Roombas. They're just endlessly patrolling the skin and the bloodstream, aggressively sucking up and chopping up this stray DNA so your immune system doesn't totally panic.
I actually love the Roomba analogy. It perfectly highlights what happens when, the battery dies on those machines. Take patients with systemic lupus erythematosus, for example. In this severe autoimmune disease, the patient's DNase activity is compromised. The shredders basically failed to do their job.
So this stray DNA just, piles up in the tissue?
It just piles up, and the immune system enters this state of chronic, inescapable alarm. It starts creating autoantibodies to attack this accumulated DNA.
Which is everywhere.
Right, so it ends up destroying the patient's own healthy tissues, their kidneys, their skin, their joints. Host DNases also chop up these sticky webs in the blood called neutrophil extracellular traps to prevent dangerous blood clots.
Wow.
So this shredding function isn't just about, cellular tidiness. It is an absolutely vital survival mechanism.
Okay, so we have this highly aggressive, highly critical defense system, but the sources break these enzymes down into a couple of different families, right? Because they don't all operate under the exact same conditions.
Yeah, they generally fall into two distinct camps. You have DNase I's and DNase the second,
Yeah.
and they have very different environmental demands to function. DNase I is a bit high maintenance, honestly.
I know.
Well, it requires what chemists call divalent cations to turn on.
Okay, wait, let's translate that for the listener. What on earth is a divalent cation?
Fair question. Basically, the enzyme needs little metal helpers floating nearby, specifically calcium or magnesium ions, just to activate and start cutting.
Got it. So it needs an accessory.
Exactly. It also only works well at a totally neutral pH. And when it cuts the DNA, it leaves behind a specific chemical signature, which is a 5' phosphate group.
Okay, so that's the high maintenance one. What about DNase?
DNase is like the rugged independent cousin. It is cation independent. It doesn't need those metal helpers at all. It actually thrives in highly acidic environments, leaves a 3' phosphate group, and is heavily concentrated in your cellular garbage disposals.
Which are the lysosomes, right?
Right, lysosomes. But it's also found right on the surface of your skin in the stratum corneum.
Which brings us to this massive, 20-year hurdle in the cosmetic industry. I mean, if our skin is literally coated in these aggressive DNA shredders, particularly that rugged DNase the second, what happens when someone goes to a department store, drops $200 on a fancy topical serum containing PDRN, and just slathers it on their face?
They're essentially just throwing their money straight down the drain.
Ouch.
Yeah, formulation chemists actually refer to this as the enzyme dilemma. You see, PDRN molecules, the polydeoxyribonucleotide, they are massive, bulky structures.
How massive are we talking?
Their molecular weight ranges from the 50 to 1,500 kilodaltons. To put that in perspective for you, anything over 500 daltons physically cannot cross the skin's outer barrier passively.
Wait, 500 daltons versus 1,500 kilodaltons? That's huge.
That's astronomically bigger.
So it literally just sits there on the surface staring at the Roombas.
Sitting right on top of the stratum corneum, full exposed. And remember, PDRN is literally just a long chain of DNA. It is incredibly fragile. Unprotected DNA polymers sitting in a water-based topical serum have a notoriously short lifespan.
So the enzymes just tear it apart.
Instantly. Those DNase and DNase enzyme stationed on your skin, they act as an immediate defensive barrier. They start snipping the phosphodiester bonds the second it touches your face. The raw PDRN in that expensive serum is completely dismantled within minutes.
It never even gets close to the living dermis.
Right, it never gets to where the collagen is actually made.
Which completely explains why cosmetic companies spent decades, I mean literally decades, trying to figure out topical delivery before finally conceding that to get real results, you have to bypass the surface entirely. You just have to inject it.
That became the only viable path. Practitioners use these ultrafine 34-gauge needles to push the liquid directly into the dermis. Usually that's about 0.5 millimeter to 1.5 millimeters deep.
But creating an injectable DNA solution isn't simple, right? Like you can't just blend it up and put it in a syringe.
No, not at all. The manufacturing standards are incredibly strict. The solution has to be hyper-purified to ensure endotoxin levels are below 0.05 EU per milligram.
Meaning there can't be any dead bacterial fragments in there, otherwise the immune system will attack the injection site.
Right. And on top of that, it has to be perfectly isotonic, hitting exactly 295 millismoles per kilogram.
Hold on, why 295 specifically? Like what happens if they just mix the purified DNA with regular sterile water?
Oh, that would be a disaster. If you inject a liquid into the skin that doesn't perfectly match the osmotic pressure of human interstitial fluid, which sits right at 295, you create a cellular nightmare.
Really? Just from the water pressure?
Yep. If the solution is too concentrated, it literally pulls water out of your own healthy skin cells and they shrivel up like raisins.
Oh, wow.
And if it's too dilute, water rushes into your cells until they literally pop.
Popping cells on a Tuesday? Not great.
Not great at all. So matching that 295 number ensures the surrounding cells stay completely happy and intact while the DNA is introduced. It's also kept slightly acidic, usually between a pH of 6.8 and 7.2, just to maintain the DNA's structural stability.
But wait, I have to challenge this whole delivery strategy for a second.
Right.
We just established earlier that these DNase enzymes aren't only on the surface of the skin, they're in the blood plasma, they're inside the deep tissue. I mean, the sources literally say tissue nucleuses are everywhere.
They absolutely are.
So if you take this perfectly balanced salmon DNA and inject it deep into the dermis, aren't you just moving the DNA directly into the path of the underground shredders? Like, why doesn't it just get chewed up instantly down there?
And that is the core of the biological tug of war. You're right, the underground shredders are absolutely waiting for it, but the actual act of injecting it fundamentally changes the physical dynamics of the encounter.
How so?
Because the injectable PDRN is highly concentrated and very viscous. It's essentially a thick, heavy gel. It forms a temporary physical depot in the tissue.
Oh, wait. Is that why people who get this treatment leave the clinic looking like they have mosquito bites? Like those little raised bumps all over their face?
That is exactly what those are.
I've always wondered about that.
Yeah, those bumps or papules are literally the physical occupancy of the thick drug just sitting in the dermis.
Yeah.
And inside that high-viscosity depot, the breakdown process isn't a flash fire like it is on the surface. It's a slow, controlled burn.
Because the enzymes can't just slice right through it.
Exactly. The tissue enzymes have to chip away at the outer edges of the gel. And this is where the physical size of the PDRN molecule becomes the single most important variable in the whole treatment. The size dictates the speed of the shredding.
Let's look at the math on that, because the sources have some wild numbers comparing the large molecules versus the small ones.
Take the large, long-chain PDRN macromolecules, the ones weighing 1,000 to 1,500 kilodaltons. When these massive tangled chains encounter the body's endogenous nucleuses, the enzymes actually struggle to get a grip on them.
Right.
The cleavage speed is incredibly sluggish. It actually drops to a rate where they only break down 5 to 7 base pairs per hour.
That's nothing. 5 to 7 an hour.
It's remarkably slow. And because of this sluggish digestion, the drug's half-life in the interstitial fluid extends to an astonishing 96 hours. Even more impressive, these large chains can stably anchor to the cellular receptors for up to 14 days.
Wait, 14 days? That is 336 hours of sustained receptor engagement.
Exactly.
It's almost like, okay, think about a giant jawbreaker candy. If you put a huge jawbreaker in your mouth, your saliva, which is essentially the DNase in this scenario, takes days to dissolve it.
Right.
But while it's dissolving, you're getting this slow, steady, continuous release of sugar over a huge span of time.
That captures the dynamic perfectly. Now consider what happens if a manufacturer tries to cut corners and uses short-chain PDRN molecules weighing only 50 to 500 kilodaltons.
Okay.
These smaller fragments encounter the exact same tissue enzymes at the exact same concentration.
Yeah.
But the rhythm shifts entirely.
Because it's not a giant jawbreaker anymore. Now it's like a handful of Skittles.
Yep, the enzymes just tear through them. The cleavage speed skyrockets to 30 to 40 base pairs per hour.
Wow, compared to 5 to 7?
Huge difference. As a result, the local concentration of the drug peaks rapidly, usually within 3 to 5 hours. And by 48 hours, the number of receptors occupied by the short chains plummets drastically.
So the Skittles are just gone, the enzymes won.
They're gone, and the cellular signal fades out. This discrepancy in cleavage speed creates a massive gap in how long the fibroblasts, which are the skin cells responsible for building new collagen, actually receive the rejuvenating instructions.
Okay, so regardless of whether we're dealing with the jawbreaker or the Skittles, eventually the DNase enzymes do finish the job. They successfully shred the foreign DNA into tiny little pieces, which the sources call oligo and mononucleotides.
Right.
But if the DNA is destroyed, why does the patient's skin look 10 years younger a month later? Like, what are those shredded fragments actually doing once they're broken apart?
Now we get into the pharmacodynamics, how the drug actually alters the body's behavior. Once the PDRN is shredded into these tiny nucleosides and nucleotides, they become the perfect key to unlock specific cellular locks.
Okay, what kind of locks?
Specifically, they bind to the adenosine A2A receptor.
What happens when that lock gets turned?
It sparks a profound anti-inflammatory cascade. For instance, the data shows that the treatment triggers macrophages to switch into what's called M2 polarization, and it increases that rate by 60%.
Oh, wait, we need to unpack M2 polarization for a second.
Sure. So think of macrophages as the body's paramedics.
Okay.
In M1 mode, they're fighting an active infection. They are aggressive. They're causing inflammation to kill bacteria or clear debris. But when they switch into M2 mode, they stand down from the attack and shift entirely into tissue repair and rebuilding.
So these shredded DNA fragments are actively telling the immune system to stop fighting and start healing.
Exactly. And even though the drug's half-life in the plasma is only about 3 hours, the healing cascade it triggers lasts much, much longer.
But the real magic, like the part of these papers that absolutely blew my mind, is the energy hack. The way the skin cells actually use the shredded DNA bits as raw building materials.
Oh, this is without a doubt the most elegant part of the biology. Let's look at the state of an older dermal fibroblast, say, in skin that's over 35 years old.
Just calling out the target audience directly, I see.
Hey, just following the clinical data. In these older cells, the internal power grid is failing. The intermembrane voltage often drops below 140 millivolts. Overall ATP production, which is the universal energy currency of the cell, falls by 30 to 40%.
That's a huge drop in power.
It is. Meanwhile, destructive reactive oxygen species surge by 1.5 times. These cells are exhausted. They're barely surviving, let alone building new collagen.
And normally, if an exhausted cell wants to build new DNA to divide and create fresh skin tissue, it takes a massive amount of energy, right? Like making it from scratch.
It is highly energy intensive. A cell relying entirely on itself to assemble new DNA from scratch consumes 6 to 8 ATP for every single molecular connection it makes.
Wow.
For an exhausted cell, that cost is simply too high. It literally cannot afford the energy bill. So the cell remains dormant. It does nothing.
But then the underground DNase enzymes shred the injected salmon DNA into monomers. And these tiny pre-cut fragments float right up to the exhausted fibroblasts.
And the cells eagerly absorb them. Because instead of building DNA from scratch, the fibroblasts use a biological shortcut called the salvage pathway.
The salvage pathway.
Right. They take these perfectly sized, pre-shredded exogenous PDRN monomers, slip them into the nucleus, and plug them directly into their own genetic breakpoints.
And here is the punchline. What is the energy cost for that salvage pathway?
It drops to just 1 ATP per insertion.
That is biological efficiency at its absolute finest.
It's incredible.
It's like, okay, imagine an exhausted construction crew showing up to a job site. Normally, if they want to build a wall, they have to dig the clay, shape the bricks, and fire them in a kiln for hours, which just takes all their energy.
Right. They'd be too tired to even build the wall.
Exactly. But instead, they show up and find thousands of perfectly sized, pre-assembled bricks just sitting on the sidewalk ready to be stacked.
The energy differential changes everything. Because the metabolic cost drops from 6 to 8 ATP down to just 1 ATP, these dormant cells can suddenly afford to resume division.
That's amazing.
Within 2 hours of the monomers arriving, cellular repair enzymes kick into high gear, rapidly stitching along the chain at a speed of 50 to 65 base pairs per second. Tissue microfissures are repaired, and fresh collagen bundles are laid down.
So let's step back and look at what this all means. The DNase enzymes, those microscopic Roombas we talked about at the beginning initially, they seemed like the villains of the story.
They really did.
I mean, they are the reason you can't just rub a fancy serum on your face. They are constantly trying to destroy the expensive injectable drugs sitting in your skin. But in reality, they are the essential preparers.
Yes.
Without the DNase chewing the DNA into those exact bite-sized monomers, the starving fibroblasts wouldn't have the pre-assembled bricks they need to use the salvage pathway.
It's the ultimate biological irony. The body's natural defense mechanism against stray DNA is entirely co-opted. The cosmetic science actually utilizes the quote-unquote enemy to act as a highly calibrated time-release mechanism. The enzymes control the pace of the degradation, ensuring a steady, long-term supply of metabolic fuel to drive intense cellular regeneration.
So if you're ever sitting in that dermatologist chair getting an injectable and you look in the mirror afterward and see those visible little bumps on your skin, you now know exactly what's happening.
Yep.
Those bumps aren't a mistake. They are your body's DNase enzymes slowly, methodically working their way through a microscopic molecular feast, carving out the exact fuel your exhausted cells need to rebuild themselves.
It transforms what seems like a simple cosmetic procedure into a brilliantly choreographed biological event.
It really does. But, you know, it also leaves us with one final, incredibly provocative thought from the source material. We know that this entire process relies completely on the host's endogenous DNase enzymes to break down the drug. Well, the research notes that DNase enzyme activity is highly variable among different human beings.
That's a crucial point. I mean, we mentioned earlier how patients with certain autoimmune conditions like lupus have severe DNase deficiencies. But even in a totally healthy population, enzymatic activity isn't uniform.
Exactly. Your shredders might work at a totally different speed than mine. Which makes me wonder, could this natural variability in our own personal DNase levels be the hidden reason why a $1,000 skin treatment yields miraculous, long-lasting results for one person but disappears completely in a few days for someone else?
Oh, absolutely.
Like if your personal Roombas are too fast or too slow, does the whole time-release system just fall apart?
It is a profound variable that the aesthetic industry is honestly only just beginning to grapple with. Because the drug in the syringe is perfectly standardized, but the patient's biological shredder is entirely unique.
It really makes you realize how much of our daily routines and our expensive beauty treatments are actually at the mercy of our invisible cellular biology. Something to mull over next time you're looking in the mirror. We'll leave you to ponder that one.